Let's learn about the next kind of passive transport, osmosis. So far we've looked at diffusion and facilitated diffusion, and osmosis is the third in this category of passive transport, meaning the movement doesn't take energy. The molecules go from high to low concentration gradient or down their gradient. Active transport is the opposite, where it goes from low to high, takes energy. That's with molecular pumps and vesicles, and those will be coming up in later videos. Passive transport is like sledding downhill. So osmosis, movement of water molecules. This is what makes it different from diffusion. This is a type of diffusion, but just a special one for water molecules. So movement of water molecules going from high to low concentration across a semi-permeable membrane. For diffusion, we don't need a membrane there. For osmosis, we do. So you can see in the picture below, the little blue circles are water. The green represents sugar. Sugar wants to go from the right to the left, but it can't because it's blocked by this membrane. So what's forced to move instead is the water. Water is highly concentrated on the left, less concentrated on the right, so water is going to go from high to low and move towards the right side. Osmosis does not require energy because it's passive. Water is the only thing that moves, and we learned about this before when I had the demonstration with the eggs and I dissolved away the shell with vinegar and I introduced certain words to you. Hypertonic, hypotonic, isotonic. That's going to come up here again. So, water moves towards the solute. I'm going to go back a slide. I always remember solute sucks. So water gets sucked where there is more stuff. In this picture you can see water is highly concentrated on the left, less concentrated on the right, so it's going to go from high to low, it's going to go towards the right. Sucrose wants to move but can't because it's blocked by the semi-permeable membrane. And again, if you think about it this way, water is getting sucked where there's more stuff, solute sucks, water is going to go from the left to the right. Now, our words, hypotonic. Hypotonic means the area that has less solute, less stuff dissolved in it. The opposite is hypertonic. So in this picture, the left side is hypotonic because there's no sucrose dissolved. On the right side, we have the hypertonic side. It's whichever has more solute concentration. So water is always going to go from hypo to hypertonic. Solute sucks. An exception to the rule is when something is isotonic. Iso means equal, and you have to look at the amount of concentration here. If you look, on the outside we have 10% salt. Inside we have 10% salt. The concentration of the solute is equal. Therefore, water is going to move in equally within the cell, without the cell. In and out equal because our concentration is equal. Let's practice here. So, back to our first example. Hopefully you still remember from before what I said. Where does sugar want to go? It wants to go left. Why can't it? Because of the membrane is semi-permeable. It's too big to get through. Where does the water want to go? Right. Can it? You bet it can. It's small enough to get through the selectively permeable membrane. So, which side is hypertonic? You have to look at solute, what's dissolved. The right side is hypertonic because there's more stuff in the water. The left side would be hypo. Another way to say this is when things are out of balance, the particles can't move because of the barrier, only water can move to compensate. Let's practice some more. The orange solutes are, let's say, sugar. And so the blue dots then are water. This makes it look like inside the cell we have 1, 2, 3, 4, 5, 6, 7 sugar molecules, and outside we have 3. So is the inside of the cell hypertonic, hypotonic, or isotonic compared to the outside? Hopefully you said hypertonic because there's more orange inside than there is outside. How about this one? Is the outside hypertonic, hypotonic, or isotonic? Think about it. The answer is isotonic. There are 5 orange dots outside and 5 inside. It is equal. How about this one? Is the outside hyper, hypo, or iso compared to the inside? Well, there's 4 dots outside, 5 dots inside, so the outside is hypotonic. Hyper, I like to think, if you're hyper, you have more energy than usual. So that's how I remember hyper means has more dots. Hypo would be less. So, how do you know which way water moves? Water gets sucked where there's more solute. In this case, the cell inside is hypertonic. Outside is hypo. Water goes where there's more stuff. It's going to go into the cell. In this case, which way is water going to go? Hopefully you said inside the cell. And just imagine water going in a cell, in a cell, in a cell. That can lead it to burst. In this case, which way is water going to go? There's 5 outside, 5 inside. Ooh, we have an isotonic solution. It's equal. So water's going to go in and out of the cell equally. Lastly, this one. Hopefully you said water is going to go into the cell. Hopefully you didn't just say outside because I didn't use that example yet. Think about it logically. 5, 6 inside, 4 outside, water gets sucked in. In terms of animal cells, our cells are happy when they're in isotonic solution. So this is a red blood cell, and water likes to move in and out equally. If you start changing the amount of stuff dissolved in your body, you could cause problems. If your cells are in hypertonic solutions where there's more stuff outside than there is inside your cells, water's going to leave and actually shrivel your cells. So this is a red blood cell that is shriveling. If your cells are in a hypotonic solution, so there's nothing really dissolved out here, much less than inside, solute sucks. Water's going to go into the cell. Here's a red blood cell that is swollen as a result. Knowing this information is actually very practical. There was a woman named Jennifer Strange, and I'm just going to summarize this article here for you, that there was a radio contest where it was called Hold Your Wee for a Wee. So they had to drink a ton of water, and if they won, if they didn't go potty, then they won the wee, whoever could last the longest. Well, Jennifer Strange drank so much water that it caused osmosis to go wrong in her body. So imagine that this is Jennifer's cells. Blue is water, the tan color would be solute. She kept drinking so much water that all of this water diluted the concentration of the materials outside her cells. So there was a lot more solute inside her cells in terms of concentration compared to the outside. Now, based on this picture, high concentration inside the cell, low concentration outside, where does the water want to go? Solute sucks. Water flooded into her cells, essentially causing lots of damage and organ failure. So drinking too much water is actually a bad thing. You're now going to do some more practice with osmosis. So you have the next page in your notes, which has pictures like this on it, plus some other ones, and you need to answer all of these questions before you come up to conference with me. To wrap up and recap before I go, we have 3 kinds of passive transport. Movement from high to low, like sledding downhill, doesn't take energy. Diffusion, just concentration from high to low. Osmosis is water moving across a semi-permeable membrane from high to low. Facilitated diffusion for anything that's too big or too hydrophobic that it can't get through the membrane on its own, so it needs its own help to go from high to low. It gets a protein. All 3 are passive, down the gradient, downhill, no energy. Next, you'll be looking at active transport and how that's different.